Okay, so welcome back to this next video in which we are discussing endothelium-dependent vasodilatation. Okay, so in the previous video what we discussed is that it is the diameter of the arterioles which determines how much blood flows to the local area which that arteriole supplies. Okay, and we discussed that there are these two opposing forces on the arterioles. There are the pro-contraction mediators, which are activating ligand-gated ion channels and G-protein coupled receptors, which both through very different mechanisms lead to rises in intracellular calcium, which then leads on to the contraction of the vascular smooth muscle cells and the constriction of the arterioles. However, those are continuously being opposed by endothelium-dependent relaxation mechanisms, okay, principally nitric oxide, but also prostacyclin and endothelium-dependent hyperpolarization mechanisms, okay, which are continuously keeping the vascular smooth muscle cells relaxed, okay, and normally, in normal vascular homeostasis, these two are all the time operating and they're all the time counterbalancing one another, and indeed, if you block one of them, for instance, if you block nitric oxide, okay, from occurring by inhibiting potentially the enzyme which creates it, okay, what you can do is cause all of the arterioles within the body to start constricting, because now this balance has been disturbed. This is no longer counter-opposing the pro-contraction mediators, and therefore you can end up with huge rises in arterial blood pressure as all of the arterioles contract, and therefore the blood ends up being trapped in the arterial system, and arterial blood pressure builds up and up and up. Okay, so it is very, very important to understand that there is this normal equilibrium, and that if you want to move an arteriole in favour of vasoconstriction or vasodilatation, what you have to do is tip this equilibrium by upregulating one or the other, basically. Okay, right. So what we now want to do is talk about how vascular smooth muscle cells contract, okay, so that we can then understand how these pro-contraction mediators are actually going to cause contraction of the vascular smooth muscle cells, and then that will give us a good grounding for then understanding the mechanisms of endothelium-dependent relaxation. Okay, right. So let's start off with the machinery within a vascular smooth muscle cell that allows it to contract then. Okay, so once again we will draw the spindle shape of a vascular smooth muscle cell here. Okay, now vascular smooth muscle cells, and indeed all smooth muscle cells, are very different from cardiac and skeletal muscle. Okay, cardiac and skeletal muscle have very, very ordered arrangements of actin filaments, okay, and myosin filaments, which gives rise to the striations of cardiac and skeletal muscle. Smooth muscle cells do not have as ordered arrangement of the actin and myosin filaments, okay, and therefore they are not striated. Okay, right, so the key thing that organises the actin filaments then in smooth muscle cells are structures known as dense bodies. Okay, so I'll draw a few of these. So dense bodies are these structures within smooth muscle cells, and some of them are within the cytoplasm of the smooth muscle cells, whilst others are just underneath the cell membrane. Okay, like so. So here we have lots of these dense bodies. So these turquoise dots, these are representing dense bodies, and these are the key organising centres for actin filaments in these smooth muscle cells. So these are dense bodies. Okay, right. So attached to these dense bodies you have a huge number of actin filaments. Now these actin filaments are generally oriented along the long axis of the smooth muscle cell, so they're generally in parallel to the long axis of the smooth muscle cell. So let me show you what I mean by that. I mean that you're going to have actin filaments attached to the dense bodies, and they're generally going to be pointing in the direction that's parallel to this long axis. Okay, so they're not going to be pointing in this direction generally, they're going to be pointing in this sort of a direction. Okay, and let me draw some more on here. This one can have some actin filaments as well, like so. This one can also have some actin filaments, like so, and then we could go on. okay, and complete the picture up. So these are the actin filaments. Okay, so let me just talk to you about what an actin filament actually is. So actin filaments are also called thin filaments. You might also hear them called the thin filaments of muscle cells. Okay, and they're made up of huge polymerizations of a protein known as actin. Okay, so actin is a small little globular protein which for our purposes we can just represent as a blob, like so. Okay, and to build it into a great big fibre known as an actin filament, what you have to do is you have to polymerize a lot of actin monomers together. Okay, what you actually do is you produce two strands of actin molecules, like so. So here's one of them, and you're going to intertwine them around one another. So let me show you this. Here is another one I'm now building, and they're going to end up intertwined around one another in an alpha helical way. I'm trying to show this carefully so that I show this one going behind this one here and then coming out in front of this one here. So they're intertwined around one another. Okay, and then this one will go like so, in this alpha helical way. So I colour this in. Here is one strand of loads of actin molecules polymerized together, here in purple. Okay, and here is another strand here in blue, and they're intertwined around one another like so. And that's what is meant by an actin filament, these two strands which are polymers of actin monomers that have been intertwined around one another in this alpha helical fashion. Okay, so that's what's known as an actin filament. So each one of these little black lines here, that's what it really is. That's what an actin filament really is. Okay, right, so now we need to talk about the other key filaments of muscle cells, which are the myosin filaments, okay, which are also called the thick filaments. Okay, and these are made out of loads and loads of myosin molecules polymerized together. So I'll show you what a myosin molecule looks like in just a moment. So the reason these are called the thick filaments is because they're much thicker than the actin filaments, and they sit in between the actin filaments like so, and they'll interlink actin filaments from multiple dense bodies, as I've shown here. So this portion is connecting to actin filaments of this dense body, this one is connecting to the actin filaments of this dense body. Okay, we could draw a few more here. And the basic idea is that when the smooth muscle cell is activated to contract, what's going to happen is the myosin filaments here, these thick filaments, are going to end up pulling on the actin filaments here, and they're going to be pulling the actin filaments towards the center of them. Okay, so these actin filaments here attached to this dense body are going to end up being pulled this way, and these actin filaments attached to this dense body are going to be pulled in this direction, and therefore you're going to move the two dense bodies closer to one another. And when you do this across the entire cell, the whole effect of this is that the entire smooth muscle cell is going to contract down. Okay, right. So what we're going to do is look at the structure of a myosin molecule in more detail, because there's something that we're really going to need to know to understand how smooth muscle cell contraction is initiated, and that's what the regulatory myosin light chain is. Okay, and without looking at a myosin molecule, it's difficult to explain what the regulatory myosin light chain is. Okay, so let's have a look now at an individual myosin molecule. Now, a myosin molecule is made up of loads of separate subunits. Okay, now the two principal subunits of a myosin molecule are the myosin heavy chains. So let me draw these now. So you actually have two myosin heavy chains in each myosin molecule. Okay, so here's one that I'm drawing here. So this is its head, and then it has an alpha helical portion like so, that will then go off like this. Okay, so this wavy line that I'm drawing here, this is supposed to be an alpha helix. And then this is the other myosin heavy chain subunit here, which also has an alpha helix portion, and these two alpha helices are going to coil up with one another, just like I previously described. The actin polymers are going to coil up to form an actin filament. Okay, so these two molecules that I have just drawn here, these are the myosin heavy chains. Okay, and for short, the myosin heavy chains are often abbreviated down to MHCs. However, that is a very dangerous abbreviation to use, because generally when people hear MHC, they do not think myosin heavy chain. They usually think major histocompatibility complex. So use that as long as you are aware that there is that risk that people won't know what you're talking about. Okay, so myosin heavy chains, these are the two myosin heavy chains. Okay, and these are both part of the larger myosin molecule. Okay, so let's draw your attention to different portions of this molecule. So this portion down here, now highlighted in blue here, this is the tail portion of the entire myosin molecule. Now, we haven't finished the myosin molecule yet, but we're not going to alter this portion anymore. We're only going to alter the heads. Okay, so this is the tail of the myosin molecule, formed by these two intertwining alpha helices of the myosin heavy chains. And again, we're not going to alter this portion of the myosin molecule here in red either. Okay, and that's what's known as the hinge portion of the myosin heavy molecule. Oh, sorry, the myosin molecule. Okay, right. So, these portions then, these are the heads of the myosin heavy chains, or indeed the heads of the entire myosin molecule. Now, they're not quite complete, because these heads are going to have additional subunits joined onto each other, and this is where we're going to meet the regulatory myosin light chain. Okay, so there are 4 extra subunits that are going to be added on here. Okay, 2 pairs, like so. So these purple molecules that I have drawn here, and we've added 1 onto each of the 2 heads of the myosin molecule here. These are what are known as the regulatory, or at least they're going to represent what are known as the regulatory myosin light chain. Okay, so MLC is used to represent myosin light chain. Okay, right. So those purple molecules are going to represent the regulatory myosin light chain, and this is going to be extremely important in the initiation of contraction in these vascular smooth muscle cells. Finally then, the little turquoise molecules that are also attached to the heads of the myosin heavy chains, these are what are known as the alkali myosin light chains. Okay, so the alkali, and I'll just abbreviate myosin light chain down to MLC. Okay, so overall, each myosin molecule consists of 2 myosin heavy chain subunits, 2 regulatory myosin light chain subunits, and 2 alkali myosin light chain subunits. So it's got 6 separate subunits to it. Okay, and then to make a polymer of myosin molecules, which is what the myosin filaments are, what you do is you join loads of these myosin molecules together. So let me just sort of show this. So I'm now going to go down to representing the entire myosin molecule just like so. This is the tail portion, this is the hinge portion, and this is where the heads are. Okay, and to make a thick filament, a myosin filament, what you effectively do is you join loads of these molecules together. All of the tail portions aggregate together, and then they have their hinge portions with their heads sticking out like so. Okay, so if I just do a few more of these. So this is the beginning of a myosin filament here. Okay, so this is what we've previously just represented as a great orange line. Now it's important to understand that when we look at these thick filaments, the portion that is intertwining with these actin filaments here and is going to pull these actin filaments in this direction is going to be orientated in the opposite way to the myosin molecules in the myosin filament that are going to be acting on these actin filaments and pulling them this way. Okay, what I have just shown you here, if this were to interact with an actin filament, okay, so if I draw the actin filament here, okay, in fact I might draw it a little bit better than that. Okay, here is one of those polymers of actin molecules, here is another one. If this was to interact with an actin filament here, it would pull it in that direction. These myosin heads will be pulling the actin filament in this direction. Okay, so how do you then get the myosin filament over here pulling them in the opposite direction? Well, what you have to do is later on the opposite side, you have to have the myosin molecules oriented in the opposite direction. Okay, like so, so that when we now have another actin filament here, these will now pull the actin filament in this direction. So at either ends of the myosin filaments, the myosin molecules are oriented in the opposite direction so that when they interact with actin filaments, they will pull them in the opposite direction. Okay, right. Now we're not going to go through the sliding filament mechanism of muscle contraction. We don't need it for what we're going to discuss. Okay, all we need to know is that when activated, what can happen is these myosin heads can interact with the actin filaments, okay, and they can pull the actin filaments in the direction towards their tails, basically. So as you can see, these ones are oriented in this direction with their tails pointing in this way. Basically, these myosin heads can pull the actin filaments towards their tails, okay? And that's all we need to understand of how contraction actually occurs. What we're going to study in a lot more detail is how you initiate the contraction. That's what we're really interested in, not the actual contraction itself. Okay, right. So, in that spirit, let's now talk about how you do initiate contraction of these myosin filaments. Well, how you initiate the sliding filament mechanism muscle contraction in these smooth muscle cells. So basically, this is where it hugely differs from what occurs in skeletal and cardiac muscle. Okay, so in smooth muscle cells, the way that you activate contraction is by phosphorylating the regulatory myosin light chains, which are attached to the heads of the myosin heavy chains. Okay, only when the myosin light chains have been phosphorylated, specifically the regulatory myosin light chains have been phosphorylated, only then can contraction actually occur. Okay, so let me introduce you then to an enzyme which is going to phosphorylate the myosin light chain regulatory subunits. So this enzyme is known as MLCK for short, and this stands for myosin light chain. Okay, so the M is for myosin, the L is for light, the C is for chain, and then the K is for kinase. So this is myosin light chain kinase. This is an enzyme which is capable of phosphorylating the regulatory myosin light chain, and when it phosphorylates the regulatory myosin light chain on these myosin heavy chains heads, then that myosin molecule can begin cross-bridge cycling and begin moving the actin filaments, and that then begins the process of contraction in the smooth muscle cell. So, what I now want to discuss is how does calcium going up inside the cytoplasm of a smooth muscle cell actually lead to the activation of contraction, which is to say, how does it lead to the activation of this enzyme, myosin light chain kinase? Okay, so the key signal that causes contraction of a smooth muscle cell is calcium level within the cytoplasm of the smooth muscle cell going up. What we now want to see is the link between calcium going up and the phosphorylation of the myosin light chain regulatory subunit. Okay, right. Let's start then with calcium going up. So we're assuming now that calcium in the cytoplasm of our smooth muscle cell has gone up. What then happens? Okay, we'll look at how calcium goes up later on. Okay, but we've said that we've got this global calcium signal where calcium is going up all over the place in the cytoplasm of our smooth muscle cell. We now want to see how it causes contraction. Okay, so the first thing that happens is these calcium ions are going to bind to calmodulin molecules. Okay, so I'm just going to tell you a little bit about calmodulin, just so that you've got a picture of it in your head. So calmodulin then. Calmodulin looks something like this. Okay, it has two main lobes, which are known as the N lobe, which can be this one here, and then the C lobe, which can be this one here. And these two lobes are connected by a linker, like so. Now, the original calmodulin molecule, which has no calcium bound to it, is what's known as the apocalmodulin molecule. And for short, calmodulin is often abbreviated to Ca, and then you put an uppercase M like that. So that's the abbreviation that is standard for calmodulin. So apocalmodulin specifically means calmodulin with no calcium bound to it. Now, how many binding sites for calcium does a calmodulin molecule actually have? Well, it has 4, okay? 2 on each of these 2 lobes. So the N lobe has 2 calcium binding sites, and the C lobe also has 2 calcium binding sites. So these little circles are representing the calcium binding sites. Now, when calcium goes up globally in the cytoplasm of our smooth muscle cell, what's going to happen is calcium ions are going to bind into these 4 binding sites of the apocalmodulin molecules. And when that happens, the calmodulin molecule changes in conformation quite a bit. Okay, so firstly, can you see that the way I've drawn this, it's sort of hunched over. The N lobe and the C lobe are sort of folded back towards one another. That's going to change. They're going to move away from one another. It's going to become much straighter. In contrast to that sort of straightening out, this linker between the 2 lobes here, this linker in turquoise here, which was initially a linear polypeptide, okay, so it was literally just amino acid after amino acid after amino acid in a straight line, that's going to take on an alpha helical structure once the calcium is bound. So what's going to happen is this apocalmodulin is going to go from looking like that to looking more like this, where the 2 lobes are now straighter. Okay, so the whole molecule has become straighter with these lobes more not bent back towards one another anymore. But the helix in between has taken on this alpha helical structure here, this spring-like structure. Okay, right. And this is when you have 2 calcium ions bound here, and 2 calcium ions bound here as well. So let's put those on. So we'll have the calcium ions shown in vivid purple. So calcium can be represented by vivid purple. And this structure now, this is what's known as a calcium calmodulin complex, or for short it can be called a Ca2+ for calcium, and then CAM for calmodulin. Okay, but I suppose I should probably write out its full name. So this is a calcium, and then it's calmodulin complex. Okay, right. So when calcium goes up globally in the cytoplasm of our vascular smooth muscle cell, it's going to bind to calmodulin, form these calcium calmodulin complexes, and then what we want to see is how is that going to lead to the activation of myosin light chain kinase. Okay, so let me just get a piece of paper then for this, another piece of paper. Right, so let's draw a picture then of what myosin light chain kinase looks like, and then we'll see how the binding of calcium calmodulin complexes is going to cause the activation of the myosin light chain kinase. Okay, right, so myosin light chain kinase then. The picture I'm going to draw of myosin light chain kinase looks like this. Here is the actual kinase domain. This is the catalytic portion of the myosin light chain kinase enzyme, which we'll have in orange here. So this is the portion that's actually capable of phosphorylating the regulatory myosin light chain. Now, the reason it's not normally active is that it has a special domain called the autoinhibitory domain, which is inserted into the active site of the catalytic domain. So this portion in vivid purple here, this is a portion of the myosin light chain kinase enzyme known as the autoinhibitory domain. Okay, and we'll see other examples of autoinhibitory domains later on. Okay, so this is the autoinhibitory domain. And for short, the autoinhibitory domain is often abbreviated to the AI domain. Okay, right. So normally this is the state of the myosin light chain kinase. It is not active because part of the protein is actually folded in such a way that it is sitting within the active site of the catalytic domain here in orange. Okay, and it's blocking that active site from actually having any function. Okay, so the catalytic domain is not capable of phosphorylating anything because its active site is blocked. Okay, so how then is calcium calmodulin complexes being formed going to activate the myosin light chain kinase here? Well, basically, calcium calmodulin complexes binds to this linker here between the catalytic domain and the autoinhibitory domain, and they change the conformation of the molecules such that now the autoinhibitory domain is moved out of the active site of the catalytic domain. So let me show this. Okay, so here now is our catalytic domain, which now has its active site free here. So I'll colour this in orange. Okay, like so. And now we're going to have changed the conformation of that autoinhibitory domain so that it's no longer folded up so that it's blocking the active site of the catalytic domain. So here is the autoinhibitory domain now firmly out of the active site. And the reason for this is that now one of these calcium calmodulin complexes is bound here. Okay, like so. So this is our calcium calmodulin complex. And I'll just put 2 purple dots on either lobe to represent the calcium ions there. Okay, so the calcium calmodulin complexes then bind to the myosin light chain kinase, and they cause the removal of this autoinhibitory domain from the active site of the catalytic domain, and that then activates the myosin light chain kinase, and these enzymes will now go and phosphorylate the myosin, the regulatory myosin light chain, and that will initiate the contraction of the smooth muscle cell by activating the myosin molecules so that they can actually perform the cross-bridge cycling. So it's essential to understand that the initiation of the sliding filament mechanism of muscle contraction is gated by the phosphorylation of the regulatory myosin light chain. Okay, now what I would also like to mention is that the way that you stop the myosin molecules from continually doing the sliding filament mechanism once they've been phosphorylated is that there is also an enzyme known as myosin lightchainphosphatase, or MLCP, which removes the phosphate groups from the myosin regulatory light chains. Okay, and this therefore reverses the work of myosin light chain kinase. Okay, so once the calcium signal is gone and the calcium calmodulin complexes have gone, then of course the myosin light chain kinase will return back to this inhibited state here, and you'll no longer have any more phosphorylation of the myosin light chain regulatory subunits, and then what will happen is the myosin light chain phosphatases will remove all of the phosphate groups that the myosin light chain kinase put onto the regulatory myosin light chains, and then of course contraction will be stopped. Okay, so myosin light chain phosphatase is responsible for removing the phosphate groups from the regulatory myosin light chains in the relaxation period of the vascular smooth muscle cell. Okay, right. So that is how a rise in intracellular calcium then induces a vascular smooth muscle cell to contract, and it's very, very important that we understand that. Okay. However, what we now need to do is fill in the other portion. We need to look at how you actually get that rise in intracellular calcium in the first place. We've looked at once you've got the rise in intracellular calcium, how it's going to cause contraction of the vascular smooth muscle cell, but we haven't seen how you actually get that in the first place. So, going back to our original picture here, we originally said that there were 2 mechanisms which were triggering contraction. The activation of ligand-gated ion channels, which led to depolarization and the activation of voltage-gated calcium channels, and also G-protein coupled receptors, which lead to liberation of calcium from the intracellular stores, principally the sarcoplasmic reticulum. Okay, so I just now want to go over these mechanisms here, okay, and again, you might be wondering, but we're doing endothelium-dependent vasodilatation, why are you telling me about all the contraction mechanisms? Well, understanding these is essential for understanding the mechanisms of endothelium-dependent relaxation, okay, so it will help a lot to understand this prior. Okay, so let's then have a look firstly at the activation of ligand-gated ion channels, which leads to depolarization of the electrical potential difference across the vascular smooth muscle cell membrane. Now, the first thing then to say is to tell you a little bit about smooth muscle cell electrophysiology. So, smooth muscle cells, like all cells of the body, do have an electrical potential difference across their cell membrane. Okay, it's quite low, it's only negative 50 millivolts, which means that the intracellular electrical potential is lower than the extracellular electrical potential by 50 millivolts. Okay, now, the other important thing that I need to get off my chest is that smooth muscle cells that line the arterioles generally are not capable of action potentials, okay? In, for instance, other tissues such as the gastrointestinal tract and the bladder and places like that, smooth muscle cells there are capable of action potentials. However, in the vascular system, we can do it as a general rule that they are not capable of action potentials. Now that makes things a lot simpler, okay, because that means that the only sort of electrical change that can occur to these cells is merely you can depolarize the electrical potential difference across the cell membrane. The only change you can do, pretty much, is just changing what the electrical potential difference is, and that can then trigger effects, okay, which is far simpler to understand than action potential, so it's quite nice. Okay, now there is a fancy word for that kind of electrical activity. People call it graded potentials, okay, because it's not an all or nothing response. Action potentials, remember, are an all or nothing thing, whereas this, you know, it's an analog system basically. It's not digital, it's not on or off. It's a case of you can depolarize it as much as you like, basically, and there are all sorts of different levels that you can have the depolarization, or indeed hyperpolarization too, and that's the origin of this being called a graded potential, that there are loads of different grades by which you can depolarize or hyperpolarize the electrical potential difference across the cell membrane. Okay, so graded potentials, not action potentials. I'll just put there, not APs. Okay, right. So basically, what you can then do is neurotransmitters can activate ligand-gated ion channels in the cell membrane of the vascular smooth muscle cells. So let's say this is a ligand-gated ion channel here. Okay, and these ligand-gated ion channels can open, and they can allow in sodium ions into the cytoplasm of the vascular smooth muscle cell. Okay, and when sodium comes into the cytoplasm of the vascular smooth muscle cell, what effect is that going to have on the electrical potential difference across the cell membrane? Well, it's going to be to depolarize the electrical potential difference across the cell membrane. Because if we think about this in terms of the electrical potential extracellularly and intracellularly, let's denote the the electrical potential extracellularly as EO. Okay, so capital E for electrical potential, O for outside, and we'll have capital E subscript I as the electrical potential inside or intracellularly. Okay, basically if we draw a nice simple diagram here, we could represent the electrical potential outside by this height, and then the electrical potential intracellularly will be lower than that. And then when you step down, when you go across the membrane from outside to inside, the amount that you go down in electrical potential is negative 50 millivolts. Okay, so when you go from here to here, your electrical potential that you're experiencing goes down by 50 millivolts. Okay, now, positively charged ions make electrical potentials higher. Okay, so if we're bringing in sodium ions into the electrical potential, into the inside compartment, then you're going to be raising the electrical potential intracellularly, because as I say, positively charged ions raise the electrical potential of the area where they are. Okay, so the electrical potential intracellularly is going to be going up. Okay, now, we didn't just get those sodium ions from nowhere, they didn't just come from nowhere, instead we took them from the extracellular compartment. Okay, so we're removing positive charge from the outside of the cell, and therefore we're reducing the electrical potential outside the cell. So the electrical potential outside is going to be going down. And I haven't really shown that going down much, but imagine that it's just gone down a little bit. Okay, I can't really show it going down that. I'll draw another line. What a mess now. Okay, so imagine that this is our new line for the extracellular electrical potential, and this is our new line for the intracellular electrical potential. And you can see now that the drop down that you will experience if you move from outside to inside is much lower. Okay, so the electrical potential difference across the cell membrane is now going to be much less negative, and the fancy word for the electrical potential across the cell membrane being much less negative is depolarization. So for instance, it might be depolarized to something like negative 35 millivolts. Okay, so the step down is now much smaller because we've moved the electrical potential intracellularly up and we've moved the electrical potential extracellularly down. Okay, right. So that's how neurotransmitters working through ligand-gated ion channels can cause depolarization of the electrical potential difference across the cell membrane of these vascular smooth muscle cells. Okay, now what does this then cause? Well, it's going to activate structures known as L-type voltage-gated calcium channels on the surface of these vascular smooth muscle cells. So let me draw this here. Okay, so from a ligand-gated ion channel to now a voltage-gated calcium channel. So VGCC is my abbreviation for voltage, that's the V, gated, that's the G, and then CC is specifically calcium channel. Okay, right. So let's add a bit of colour on there now, so that we can distinguish between the ligand-gated ion channel and the voltage-gated calcium channel. So here's the ligand-gated ion channel in red. Okay, here is the voltage-gated calcium channel in orange. Okay, so these voltage-gated calcium channels, they are activated by depolarization of the electrical potential difference. So once you've depolarized the electrical potential difference across the cell membrane up to negative 35 millivolts, these things are much more likely to be open. Okay, so a good number of them are now going to open, whereas they were previously closed. Okay, and that means that we're going to allow calcium to come in from the extracellular fluid into the intracellular fluid. Okay, so whenever you open a voltage-gated calcium channel, you always get calcium moving in, because calcium concentration extracellularly is much, much higher than it is intracellularly. The intracellular calcium concentration is usually around 100 nanomolar. Okay, that's intracellular calcium concentration, whereas extracellular calcium concentration is around 1.5 millimolar. Okay, that is a 15,000-fold gradient of calcium across the cell membrane. Okay, so whenever you open a voltage-gated calcium channel, you're always going to get calcium coming in as the net movement, simply because a calcium ion is 15,000 times more likely to hit the ion channel from this side and go through this way than it is to hit the ion channel from this side and go this way. Okay, so you're always going to get a net movement inwards. Right, okay, so you get calcium coming in through these voltage-gated calcium channels, and then that causes this calcium signal. Okay, now, I just want to quickly explain what is meant by an L-type voltage-gated calcium channel, okay, because these voltage-gated calcium channels in vascular smooth muscle cells are specifically L-type voltage-gated calcium channels. Okay, so, basically, to do this, I just need to tell you a little bit about the structure of voltage-gated calcium channels. Okay, so unfortunately, voltage-gated calcium channels are not just one protein. They are a complex of proteins. So let me draw this in the cell membrane here. So let's say this is now representing the cell membrane of our vascular smooth muscle cell. This is representing the outer leaflet of the cell membrane. This is representing the inner leaflet. Okay, so I promoted the membrane from being represented just as a single line to being represented as a bilayer. Okay, so the main protein that makes up a voltage-gated calcium channel, the principal, the most important subunit, is what's known as the alpha-1 subunit, which is this subunit that I've shown here. Okay, so let's colour this in. This is the one that actually has the pore going down the middle that allows calcium to move through, and it is actually just a single protein. Okay, it's one massive great protein. Okay, now, voltage-gated calcium channels also have a bunch of other subunits attached onto their alpha-1 subunit. So let me show these now. So for instance, you also have a gamma subunit sitting next to the alpha-1 subunit here. Okay, and I'll colour that in blue. You also have a beta subunit that attaches to the underside of the alpha-1 subunit down here. Okay, so this is our beta subunit. And then you have a weird subunit over here called the alpha-2 delta subunit. So this is the delta portion here which crosses the membrane, and then you've got the alpha-2 portion up here. Okay, and that sits alongside the alpha-1 subunit here. Okay, now the alpha-2 and the delta portion are combined together as the alpha-2 delta subunit, okay, because they actually are made by the same precursor protein. Okay, so let me just explain this. So there is a gene for the alpha-2 delta subunit. It makes mRNA, which then makes the protein, which is an alpha-2 delta pre-protein. Okay, it's the precursor protein. This is then cleaved into two separate proteins, the alpha-2 subunit and the delta subunit, but they then attach back together in a different way to the way they were originally attached. Okay, so originally they were attached by peptide links. Okay, they're not attached by peptide links anymore, but because they come from the same gene, that's why they're often clustered together as a single subunit known as the alpha-2 delta subunit. Okay, so that's just to explain why you consider these two as a single subunit. Okay, because you might be wondering, well, these are all attached to each other. Why have you considered those two as a single subunit merely because they're attached to each other? Well, it's because they came from the same gene, basically. Okay, right. Now, the other subunits all modulate the function of the alpha-1 subunit. Okay, they, for instance, affect where it will actually be localized within a cell. Okay, but the alpha-1 subunit is the most important subunit fundamentally. Okay, now, there are 10 different alpha-1 subunits that you can use to build a voltage-gated calcium channel, and it is this that affects how you name the voltage-gated calcium channel. So let me tell you about the 10 different alpha-1 subunits. Okay, right, so the 10 different alpha-1 subunits are clustered into 3 families, and they're named CAV1 family, the CAV2 family, and the CAV3 family. And let me just move this up. Okay, so CAV stands for voltage-gated calcium channel, and then this is the first family, the second family, and the third family. Now in the first family you have 4 members. Okay, you have CAV 1.1, the first alpha-1 subunit. You then have CAV 1.2, CAV 1.3, all the way over to CAV 1.4. Now, if you use one of these alpha-1 subunits in the CAV1 family to make your voltage-gated calcium channel, then your voltage-gated calcium channel is known as an L-type voltage-gated calcium channel. So that is specifically what is meant by L-type voltage-gated calcium channel. It means that the alpha-1 subunit that you used to construct that voltage-gated calcium channel was in this CAV1 family of alpha-1 subunits. Okay, now for those who want to know, the specific alpha-1 subunit that you use in vascular smooth muscle cell L-type voltage-gated calcium channels is actually CAV 1.2. Okay, so these specifically are CAV 1.2 L-type voltage-gated calcium channels. Okay, now just to complete the picture of voltage-gated calcium channels, to finish what I started, the other 2 families each have 3 members as well to take us up to 10 total. Okay, so there is CAV 2.1, CAV 2.2, CAV 2.3, and there is also CAV 3.1, 3.2, and 3.3, which takes us up to a total of 10 alpha-1 subunits of voltage-gated calcium channels. Okay, so the other 2 families are not important in vascular smooth muscle cells, or at least are not believed to be important. Okay, right, so the voltage-gated calcium channels then open, and that allows calcium into the cytoplasm of the cell, and that's what creates a global calcium signal that then causes the contraction of that vascular smooth muscle cell. Okay, right. So we will call it there for this video, and in the next video what we'll turn our attention onto first is the G protein coupled receptors and how they can produce a global calcium signal through the mobilization of intracellular calcium stored in the sarcoplasmic reticulum. Okay, and then we'll turn our attention onto the endothelium dependent relaxation mechanisms.